everyone. So again, my name's Hillary. I'm an epidemiologist, and I'm coming to you from I-ARC. And I learned sort of in the pre-dinner pool meeting last night that I should, in fact, explain what I-ARC is. So I-ARC is the International Agency for Research on Cancer. We are located in Lyon, France, and we're part of the World Health Organization. So the mission of I-ARC is to promote international collaboration in cancer prevention research. And at I-ARC, I co-lead the risk assessment and early detection team. So I'm sorry to tell you that you have to listen to me for sort of a while because I'm going to give my own talk and then I'm going to give a talk in place of Chris Amos. So the first one will address the question of biomarkers, how good are they now? And the second one will address how good do they need to be to make them useful. And in the second part, I'm going to tell you about our own research as a way to kind of talk you through how we think about that question. But for the first talk, I'll try to address how good are they now. Let's see. Okay. Nothing I'm about to say is going to be groundbreaking for this group, but just to address kind of how we think about blood biomarkers. And I forgot to say, the original title of this talk was Image and Blood. I made an executive decision to focus only on blood because we've talked a lot about imaging biomarkers and you all know much more than I do, so I'm going to talk about blood only. When we talk about blood-based biomarkers alongside lung cancer screening, really what we're trying to do is optimize risk assessment at each decision step. So, of course, the first decision step is for an individual, should screening be initiated? And we can use kind of broad criteria like PACYER's task force criteria. We can improve the efficiency of screening by replacing those criteria with risk prediction models. So same number of people screened, more cancers detected. And what we want to do is improve further beyond that. But if we're going to improve further, we have to integrate novel information into those risk models. And in my view, the most likely source of this novel information is from biomarkers. So this is the idea. For a person who is high risk and goes on to be screened, we now have a lot more information because we have a low-dose CT scan, and we can apply AI, for example, to that low-dose CT scan. But again, we want to improve our risk assessment for that individual person so that we can make better decisions. Those decisions could be, how should lung nodules be managed? How long should the screening interval be for people who are free of nodules? And yesterday, I think it was yesterday, Matthias laid out sort of for a radiologist looking at an LDCT image, there's a series of sort of steps. There's a series of decisions that radiologist has to think through. And in the same way, the AI needs to walk through those steps in that order as well. And the case is sort of the same here for biomarkers. We have to think about what is the task at hand and can the biomarker achieve it? In my group, we're not usually thinking about these two questions, but at this particular meeting, we are thinking a lot about these questions. So could we use biomarkers to think about whether the participant is impacted by coronary artery calcification or emphysema? And tomorrow we're going to discuss for early stage lung cancers, can we sort of distinguish between those that are maybe overdiagnosed, those that are sort of well-behaved, and those that are more aggressive than we thought. Luis Sejo, a few years ago, 6 years ago, apparently that's horrifying, put together a really nice review of biomarkers that have been proposed for use in lung cancer screening. This figure is still very relevant. So there's different sources sort of in the body of where we can take these biomarkers from. There's of course blood, but also we can use, for example, urine, we can use exhaled breath, and we can look at many different analytes in those samples. So we can analyze DNA mutations, microRNAs, metabolites, proteins, autoantibodies, and so forth. So there's really quite a broad spectrum of biomarkers that we could consider. I was asked a few years ago to give a talk on who could benefit from biomarkers in lung cancer screening, and to sort of make this perhaps overly simple. When we apply a biomarker, if it provides useful information, then we either learn that the person is higher risk than we thought before, or we learn that the person is lower risk than we thought before. So for people who are higher risk than we thought before, they might not be eligible for screening, but they could become eligible based on the biomarker. They could receive nodule workup more quickly or more aggressively than we thought was needed. or they could have lung cancer that is more aggressive than we thought it was going to be based on the information we had about the stage. For people, on the other hand, who are lower risk than we thought they were before, they might be eligible for screening, but perhaps they can avoid it because the biomarkers are telling us this person is not going to develop lung cancer. Perhaps they can avoid nodule workup, and perhaps the lowest hanging fruit is that they could lengthen the interval between their screens. So where do we stand with blood biomarkers? I'm going to give you a bit of a rapid review of a few different biomarkers that have been proposed for use in lung cancer screening. It is absolutely impossible to be comprehensive in doing this. And similarly, I am not an encyclopedia of biomarker research. My goal is to kind of tell you what's out there and how we should think about it. So I'll walk through a few examples. The first is the microRNA classifier, which was evaluated in the BioMild study. So in the initial publication from a few years ago, what we saw was that among those who were CT negative, I think this was at baseline, there wasn't really much difference in lung cancer incidence between the groups that were positive and negative for the biomarker. So those are the two curves at the bottom. What was really quite remarkable is that the the double negative group, so the CT negative, biomarker negative group, was referred to a 3-year screening interval with no apparent important consequences of doing that. So this was actually quite innovative. But for the biomarker, there was a big difference between the CT positives who were positive and negative for the microRNA biomarker. So at the time we thought, okay, this biomarker doesn't seem to do anything for the CT negatives, but it seems informative for the people who had a nodule. So just last year, the same group published longer-term follow-up among the about 1,000 participants who had a nodule at baseline. And indeed, they found that there was an 8-fold increase in lung cancer mortality for the people who were positive for the microRNA classifier compared with those who were negative. And this increase was robust to adjustment for the PLCO score and for the BROC score. So this is quite interesting data, and I think it remains to be seen exactly how to implement such a test in the context of nodule management that's already a bit complicated, but it's promising. So another kind of domain that I work in is multi-cancer early detection. So these sort of novel multi-cancer tests. And we're not talking about that at this meeting, but I think it's worth addressing because it's sort of in the ether where all of us are sitting. So the perhaps most discussed of these multi-cancer tests is the Gallery test, which was developed by Grail. And if we focus, kind of zoom in on the story about lung cancer, from their case control data published in the CCGA study in 2021, we can see quite clearly that the problem this test has for lung cancer is that the sensitivity drops dramatically when we look at stage 1. So the sensitivity for lung cancers these are samples collected after diagnosis of symptomatic stage 1 lung cancer. Symptomatic is a loosely used term. The sensitivity is 22%. So this is already a bit concerning for lung cancer. This test was then evaluated in a prospective interventional screening study of about 6,600 people called Pathfinder. In Pathfinder, there was a single test at baseline with Gallery and then a 12-month follow-up. And among the cancers that were diagnosed in this group, only one of them was lung cancer, and this was a stage 3 adenocarcinoma. So again, I don't think we see a lot of evidence that a test like this can add something when we have LDCT, which is a test with extremely good performance characteristics. I'll tell you about a second multi-cancer early detection test. Gallery was not the first to roll out a large interventional screening study. CancerSeek was the first. And in their initial retrospective case control data, so analogous design, they found a higher sensitivity, if I calculated it correctly, for stage 1 lung cancer of about 51%. And in line with that, so CancerSeek was evaluated in a study called DETECT-A, which was published in 2020. This was an interventional screening study of 10,000 women, and it had basically the same design, single test, 12-month follow-up. And here there were more lung cancers identified. 9 lung cancers were identified in these participants, but only 2 of these were treated by surgery with intention to cure. So again, I think we have to be cautious in thinking that these multi-cancer tests are going to have an important role when we have such a good test for lung cancer screening already. Another test is the early CDT lung autoantibody test. And this, again, is a bit of an interesting story. So my team actually evaluated the performance of this test in the consortium that we organize, which I'll tell you about in the next talk. But the important thing to know about the Lung Cancer Cohort Consortium is that these are pre-diagnostic samples. So these are blood samples collected not after diagnosis, but before diagnosis. And when we looked at the performance of this early CDT autoantibody test, we found a sensitivity of 14%, a specificity of 86%. And if you add those two together, you get 1.0, which means that this test in our data was equivalent to flipping a coin with a probability of 14%. The early CDT test was evaluated in a randomized study in Scotland, which was designed as follows. Participants were those with 20 pack years. They were older, 50 to maybe 75, something like this. And they were randomized either to an intervention or control arm. In the intervention arm, everyone received the biomarker test. If it was positive, they were referred to low-dose CT screening. In the control arm, they did nothing. So no biomarker, no screening. And at the end, they compared the lung cancer stage distribution between these two arms. So what would you expect to see in this stage distribution? You would expect to see more early stage lung cancers in the intervention arm, because why? Some of these people received low-dose CT screening. Does that mean the biomarker worked? No. Even if the biomarker is a coin flip, when we screen some proportion of people with low-dose CT, we will get a better stage distribution. So unfortunately, we can't actually distinguish in any which way the benefit of the biomarker from the benefit of low-dose CT screening in this study design. So ultimately, the NIHR in the UK reviewed this test, the early CDT test, and they concluded that it has poor diagnostic accuracy, low sensitivity to detect malignancy, and it's unclear what it adds to existing diagnostic methods. So I think, again, a bit of a cautionary tale. We have to be really careful that we understand what a test is doing and that we can learn something about what the test is doing based on the studies that we design. All right, last one. This is referred to as the 4-marker panel protein assay. And this 4-marker panel was developed, it's being studied by a group at MD Anderson. And in a 2018 study, the group compared the performance of their 4-marker panel algorithm to the PLCO M2012 model in samples from the PLCO study. So these are appropriately pre-diagnostic samples. And they found that the AUC improved from 0.8 for the PLCO model to 0.85 when they applied the 4-marker panel protein algorithm. Last year, they took a next step, which was to look at the utility of repeated measures. And this is really a useful and interesting thing to pursue, particularly with protein markers, because protein markers change. They can change quickly within the same person, as far as we can tell. So they developed an algorithm to analyze repeated measurements of the 4-marker panel within the same person, and they compared that to what they got if they only measured the proteins once. And they were able to show an improvement in AUC from 0.81 for a single measurement to an AUC of 0.86 for the repeated measurements. So this is, I think, quite an interesting assay as well. But there's a few important things to note that are a bit under the hood for this evidence. The first is that for the repeated protein algorithm, this is basically internal validation. So it's not an external validation of the repeated measurements algorithm. And maybe more importantly, in the PLCO, which is where their data are largely based, the controls that they selected were required to be free of all types of cancer all the way through the entire 13-year follow-up of the PLCO. So when we're talking about older people who were enrolled in the PLCO study, you can imagine that a very high proportion of them would develop some type of cancer over these 13 years, which creates quite an important selection bias when we have the lung cancer cases and the controls that they were ultimately compared to. So what this basically makes is an easier problem for the biomarker, because the biomarker is trying to sort out people who develop lung cancer from people who were sort of unusually healthy because they didn't develop any type of cancer. It's a bit of a sticky concept, but again, it means that if we wanted to understand exactly how this panel would perform in the population in which we intend to use it, we would need to do another study. All right, so to sum up, blood-based biomarkers in lung cancer screening, how good are they now? My view is that we have not identified any unicorn biomarkers yet. Another domain that I work in is early detection of HPV-driven cancers. In that field, we have unicorn biomarkers. We have really incredible biomarkers with really long lead time that sort of tell you exactly who's going to develop oropharynx cancer, for example. That's not the story we have for lung cancer at the moment. But there are promising results for different domains of markers, and I think these include proteins, they include microRNAs. But clearly, to study these, we have to clearly define the clinical application that we're aiming towards, and we have to design studies in a rigorous way in accordance with that clinical application. So I'll stop here and take questions. Who's in charge?
Are there any questions? What about the volatile organic compounds in the exhaled breath? I was working on that for several years. Do you believe that is something?
I think it's coming along, but we have less. I'm not an expert at this domain, but we seem to have less evidence about how they perform compared with the blood biomarkers for the moment, which is why I sort of didn't include them. But there's, I think it's an area where technology is going to help us a lot. So I think it remains to be seen kind of where it's going. Maybe you can comment.
Okay.
Fred, can I ask a question?
Sure.
Perry, great talk and a wonderful overview. Do I take the conclusion is that actually looking forward, we really need to test biomarkers in prospective screening studies? CT screening has improved so much since the era of, let's say, PLCO and LST, that in fact, we need to make sure the biomarker is actually contributing something to the impact of CT screening today. So in fact, to show that a biomarker is going to be effective, in my view, we have to actually look at current screening studies. I mean, the Targeted Lung Health Check, maybe one in the UK, other European studies across the board. But I think we need to be collecting specimens now to actually test the future biomarkers.
Yeah, thanks, John. I absolutely agree with that. We need to be collecting specimens now. We need to be collecting them from as many people as we can at as many steps of the screening process as we can. We need to process them in as many different ways as we can afford. We need to store them in as cold of a freezer as we can afford. We need to try to be systematic to avoid sort of bias in the populations in which bloods are collected. So when you use the word perspective, indeed, I think this is critically important because these samples would be pre-diagnostic. They provide a proof of principle that we could have identified lung cancer earlier. Once somebody's diagnosed with lung cancer, you collect blood at that point. You can learn something, but you can't learn as much. So I agree with what you're saying. And I guess the last thing I would add is that the studies don't necessarily need to be interventional. Yeah, you can learn an enormous amount, even if you don't use the biomarker to change your decision making. I think at the end of the evidence pathway is that, but there's a whole lot to do before you get there.
I have a comment on this before. The reason we learned about biomarkers and treatment in advanced disease was because we learned about the biology of the tumor first. We were involved very much in the EGFR story, and you need to take the tumor tissue. So my point is, we need to learn more about the biology of the nodules, screen-detected nodules, before we can expect to have a good biomarker for blood, for example. I think we need to learn the biology of the tissue. David, I apologize for.
I think that area is very blurry, when you're looking for DNA fragments or circulating tumor cells and they talk about a risk of cancer in the future, this gets really, I don't know. I think there's just a lot of confusion in this area. What is it that the markers are measuring? And are they markers of risk or markers of for diagnostic purposes? And somehow these get confused. And I think that that's such a critical issue. And as I said, some of the, I would have thought were diagnostic, much more for the diagnostic purposes rather than for risk purposes. So I'll be very curious to see how you sort that out.
I think we will have that discussion in a panel discussion. Otherwise, we need to, go ahead.
Maybe just a quick one, and I think it correlates with what David just mentioned. Yesterday, we had a discussion on the CT capable of providing information of very small nodules. Well, quite obviously, those potentially malignant can produce some form of biomarkers. But then the discussion was that below a certain size, well, we don't really do anything about that. Then I think the major dilemma that is going to come from those studies is it is positive on biomarker and it is negative on the CT. What do we do? Because in everything that you demonstrated so beautifully, we still use the low dose CT as the tool to measure sensitivity. And then it's a kind of like, I see some discrepancy here that I think would be interesting to explore further.
Yeah, exactly. The answer to your question is it depends how good the biomarker is and what we know about what it means. And to give a 2-second answer to David's question, a risk is something that we calculate. Yeah. So no biomarker is perfect. No biomarker has 100% sensitivity and specificity, which means that underneath is a probability. And that's the risk. Even if biologically the biomarker is either responding to a cancer or telling us about something happening in the long term. I'm going to make it more clear, I hope, and we can talk about it in the panel.
Do you have another talk? Or?
Yeah, I'm giving Chris's talk.
Okay. Oh, okay. Yeah, please go ahead. Will you introduce Chris or?
Chris isn't here.
No.
Chris, okay, so Chris Amos is, oh, this is funny. I'm completely unprepared. Chris Amos is the PI of the Integral U19 collaboration that we're working within, which is why I work with Chris. And he does a lot of germline genetics research on lung cancer, basically. He's just moved from Baylor to University of New Mexico, which has thrown our entire U19 into chaos, but that's a story for another day.